everyone, welcome to Study with God. In today's video, we are going to do the part 2 of structure of chromosomes, cell cycle and cell division. The topic for the day is the histone protein. Chromatin. Chromatin was divided into two, DNA and histone protein. DNA is 40% and histone protein is 60%. Today we are going to cover the histone protein as DNA was covered in the earlier video. So let's start with the histone protein. Let's talk about histone protein. Now what is the function of histone protein? The function of histone protein are, it helps in packaging and coiling of DNA. Into structural units which are called as nucleosomes. Nucleosomes. Clear? So what is the function of histone proteins? It helps in packaging and coiling of DNA, and these two are keywords. Not gonna miss any of the keywords in this video or any other video, the further videos of mine, and you are also not going to miss any keyword in your exams. Okay, I'll try my best for this. Now talking about this nucleosome, in the previous video you have learned about nucleotides. So ये तो 1 मार्क्स का आता है. अगर न्यूक्लियोसोम को सिंपल वर्ड में बोला जाए तो it's one DNA strand. It's composed of one DNA strand and 8 histone molecules. This is nucleosomes. The another word which is nucleotide sounds similar. This comes for differentiate, and in Half-Yearly and that is in the board exams and in any of the pre-board it definitely comes for 2 marks or separately 1 marks each. So let's do the difference between these two words. So let's do the differentiate, and with the help of this differentiate you will able to recall this nucleotide wala concept. Okay, so let's do this. What are nucleosomes? Nucleosomes compose of one DNA. DNA stands for deoxyribonucleic acid, spelled as DEOXYRIBONUCLEIC acid, deoxyribonucleic acid. It also comes for 1 marks in expand, and even ribonucleic acid, which is RNA, which will further study about it. This also comes for 1 mark, so you have to remember these two things. Okay, so composed of one DNA strand and 8 histone molecules. Achha, one more thing. In exams or in any of your tests and all, you have to write the full form of DNA. Please just don't write DNA like what I have written. Even though I have written the full form, you also write the full form. Okay. And let's come to nucleotides. I think even I was at the starting or the beginning I was also confused between nucleosomes and nucleotide क्योंकि इतना similar लगता है. चलो अब आते हैं nucleotide के बारे में. So these are repeated units on DNA. What? Or you have to make sure that it is on DNA. There is deoxyribonucleic acid made up of, I'll show you the diagram also. Made up of pentose sugar inorganic keyword, phosphate and nitrogenous bases. Now, talking about nitrogenous bases, it's out of 4 types: adenine, thymine, cytosine, and guanine. DNA so that I can tell you what are nucleotides. Even though this I have covered in my first part of this chapter, you can also watch that. So I'm not going that deep in this video, sorry about this, because this I've already told you. So this is one nucleotide, this is your inorganic phosphate, this is your pentose sugar, and this is your nitrogenous base. Actually I've only made one. After that topic, the one more important thing comes, that is rearrangement. So let's do that rearrangement. That rearrangement is DNA, histone. Now I'll tell you one more thing over here also. Nucleosomes, chromatin, fibers or chromatin, whatever you want to say. Actually chromatin fibers, and chromosomes. Sometimes it comes ना कि first word को देख के underlined होगा, उसके बाद बाकी आपको serial में लगाना है. Over here it can come over here also, and it can also come over here. अब इसमें जो आपको underline मिले, whatever you get underlined, from there you have to start. Each chromosomes consist of sister chromatid, centromere, and non-sister chromatids also. So this part is called as centromere. The बीच में question, I will tell you the questions also which will come. So centromere is the point of attachment of sister chromatid. What are sister chromatid? These are your sister chromatid. This is centromere. Centromere, point of attachment of two sister. This is first sister chromatid. This is your second, or first second. You can take two sister. This is the centromere. Okay. Now let's do a difference between centromere and chiasma. So this is one chromosome. If I make one more chromosome and I do this, ये आप आगे वाले chapters में पढ़ोगे. Actually मैंने आपको अभी बता दिया. This is one chromosome, this is other chromosome. This was centromere. ठीक है. This point where two non-sister chromatid, एक ये अपनी अलग है, ये अलग है, तब एक इसका और इसका तब जुड़ता है. These are non-sister chromatid. इस point को कहेंगे chiasma. Clear? So इसके और इसके बीच में difference आता है 2 marks का. Centromere, point of attachment of sister chromatid. Chiasma क्या है? Point. Oh wait, let me write it in a different way. Should I write it or write it? Chiasma. Point of attachment of two non-sister chromatid. Okay, so this counts for difference. Now let's come to genes. Now talking about the genes. Genes are specific sequence of nucleotide. Now till now you should know what are nucleotides. On our chromosome, it is on a chromosome. Okay, if normal location, this is the definition of a gene. But if normally it is asked what is the location of gene, so the location of the gene will be till here on a chromosome. So this will act as a locations. Okay, that encode particular protein which express in a form of a body feature like your hands, your body feature like a face, whatever, the eyes and all. like कहते हैं ना कि इस पे need for your new cells. The new cells are need for repair, replacement, growth, reproduction. When you fall down, you get a hurt over there, you need new cells. Hope you like today's video. Please like the video, share it with your friends, subscribe to my channel, and please press the bell icon. And please watch the third part, which will be coming very soon, which will be on cell cycle. So bye bye.